Hello, my name is Alyce DeMarias, and I am Professor Emerita at the University of Puget Sound Department of Biology. I'm delighted to be part of the Science Stories project and to work with the wonderful artist Jessica Spring. I am also delighted to have been a part of biology at the University of Puget Sound for many reasons, but one being that I could work with talented undergraduates in the research lab. And much of what I'm going to talk about today was developed by these students. These undergraduate students have led our research projects to the idea of looking at the effects of environmental chemicals on egg development. And we're interested in how those eggs are forming in the ovaries of different organisms. And in particular, we're looking at the effects of the chemicals involved in polycarbonate plastics because those plastics are so ubiquitously used around the world. And this graph shows that the market value just in the US for these types of plastics only is going to increase as we continue in the next several years. One of the reasons we're interested in these environmental chemicals that are found in plastics are that they are very similar in structure to the steroid hormone estrogen. And estrogen is a hormone that is particularly important in egg development in the ovary. The chemicals that we've been looking at are bisphenol A and its relative bisphenol S. And you can see from these structures that these chemicals do have features that are very similar to estrogen. This makes these chemicals endocrine disrupting chemicals, endocrine because they're mimicking a hormone that's found in the body. And these happen to be estrogen mimics. We're interested in both BPA and BPS because as BPA is becoming more studied and the negative effects of it are known, BPA is being banned in a number of countries and in a number of products. And so the use of BPA is going to decrease over time. And BPS is a helpful surrogate for BPA. So BPS can be used in place of BPA, still giving us those chemicals. So why are we interested in these in egg development? Well, as I mentioned, estrogen is an important hormone in egg development. If you have an egg here called an oocyte in the ovary, that egg will respond to an estrogen signal, and that signal can allow the egg to go in one of two directions. One is if the egg isn't ready to be released or ovulated, that egg might go to cell death, which is known as apoptosis. If the egg is going to be ready to be developed further, it might go through cell division. And so we have this nice balance between apoptosis and cell division that are important processes in the formation of eggs. And we are interested in particular proteins or genes that are involved in this process. And if you're looking at apoptosis or cell death, caspase is one gene that's involved in that process. And beta-catenin is a gene that's involved in cell division. And in the middle here, we have a director, P53. P53 is a protein, a gene that codes for a protein that can help direct that process to either cell death, apoptosis, or cell division. And so those are three different proteins that we've been interested in looking at. How we look at those is we use a model system, and zebrafish are small aquarium fish that have become an important model system for vertebrates and the physiology and anatomy and development and just a living being of most vertebrates. And so we can use zebrafish as this model. We can study them in the lab, and we can make inferences about what might happen in other animals, other vertebrates, including humans. And to do that, we take the ovary tissue from these zebrafish. We can put them into the lab. For example, in this dish, you can add chemicals to the ovary tissue in the dish, you can wait a little while at certain amounts of incubation times, and you can look at them under a microscope, or you can look at the RNA and the proteins, and that's what we did mostly. So here again is our idea that we have two pathways that are directed by the protein p53. So there's p53 and there's beta-catenin that can lead to cell division, and there's caspase that can lead to apoptosis or cell death. And so here's a cell that is in its kind of happy normal state. If it is going to divide to make two cells, it will go through the cell division process. And if it's going to go through the cell death process, it becomes nice and packaged and essentially recycled. Caspase, if it's turned on, that protein will lead to apoptosis or cell death. Beta-catenin, if it is turned on, will lead to cell division. Beta-catenin will stop p53. p53 will start caspase. So this makes this very fine-tuned, balanced. What the students have found in doing this work in the lab is similar to the following. So don't be afraid of this graph. This is here to show you some of the data that we receive when we do this type of work. Here is on this axis the amount of protein expressed, or the amount of RNA expressed, which would lead to protein. And on this axis, we have 4 different treatments. We have no BPA, we have a very low level of BPA, 0.01, and here we have a moderate level of BPA, 0.1, and here a higher level of BPA, 1. And if we look at the response compared to no BPA, we see an increase in the amount of RNA, which would lead, we think, to protein at the low levels of BPA, that then decreases as we increase the exposure to BPA. This happened to be the gene that was in, or the RNA that was involved in producing caspase. So remember that caspase was that protein that was involved in cell death. And so if we have low levels of BPA, we see an increase in that protein, and we're going to infer that we have increased levels of apoptosis or cell death in that response. So that's one example of some BPA exposure. We can do a similar experiment with BPS using the same concentrations. And here is, and looking at caspase. Here again is our amount of RNA that's being produced. That's going to, we think, lead to protein. Here we have the same 4 concentrations, but now of that chemical BPS. So no PPS, very low BPS, an intermediate amount of BPS, and a higher amount of BPS. And I hope you notice when you look at this graph that it's the opposite effect of what we saw with BPS, or excuse me, BPA. So at lowest levels, we saw a decrease in caspase expressed rather than an increase. And so this is obviously a very complicated system, and it gets more complicated when we look at different proteins. So here is work on the protein p53 with BPS and looking at how it is compared to estrogen. And we have similar graph. We have the amount of protein that, or the amount of RNA produced on the axis here. And we have different concentrations here. And in this case, we see if we just compare BPS with similar amounts of estrogen, then we see different responses. So we see a U-shaped response with BPS and a decrease at the same concentrations with estrogen. And these are called non-monotonic responses. They are not linear. So the main thing to think about with this is that we're seeing different effects of different chemicals on the genes that are involved in egg development, and we are seeing responses that aren't linear, that they may go up and then down. So the summary of a lot of work that we've done in the lab is illustrated in this busy slide. And so again, I'll point out that we have our three proteins that we're interested in: beta-catenin, p53, that director, and caspase. That beta-catenin is involved in cell division. That caspase is involved in apoptosis or cell death. Now, I didn't show you any of the data with beta-catenin, but we have seen that low BPA levels actually decrease beta-catenin. And so that would send the ovary cells over into this direction. But low BPA increases caspase. So we have a disproportionate amount of caspase being produced with low BPA and less beta-catenin. So you could infer from that that we have more apoptosis with low BPA in the ovary. Low BPS, on the other hand, decreases caspase, decreases apoptosis, and we've seen that that balances as well with cell division, increasing cell division. So we've got this two different proteins, caspase and beta-catenin, thrown p53 in there as well, two different chemicals that are related, BPA and BPS, showing different effects on the ovary. What that tells us is it's a complicated picture, and it tells us that the ovary cells are responding to these environmental chemicals, these endocrine disrupting chemicals. And this fits into an interesting and complex relationship that we have with the environment and with the different products that we make as a culture. And it really draws to mind the poem by Percy Shelley that Jessica Spring has used for her artwork. And one line in the poem in particular that has struck me is this idea that nothing in the world is single. And that really fits well with what we've thought about in studying these environmental chemicals and their effects on egg production in specifically. So we have the natural system with our physical environment and our biota, our animals and plants and microbes and archaea. And we have the raw materials and energy that we use so well as humans. And we have our social systems, our institutions and our culture and our economics that are so tied to our production of things like plastics and our social system outputs of waste and interventions, things like BPA that come out of those chemicals. But all of these things are influencing not only our environmental integrity, but our robust, resilient economy and our health and well-being. And we want to know how that happens. And one of the things that we really are working with when we think about this in the lab is that it's not a cut and dried bad and good situation. So we can say that BPA and BPS and similar chemicals affect egg development, but where would we be without them? We certainly wouldn't want to have a medical system, for example, without these types of products. And we wouldn't be able to transport water in a very efficient way if we didn't have these products as well. So where does the balance come in? And I think that's really captured beautifully in Jessica's work, where she's juxtaposed this amazing poem with the thoughts about water and natural environment and the effects of that and the influence of that on humans and how we interact with each other. So I hope you enjoy the Science Stories exhibits, Jessica's work, and our work with the BPA and BPS chemicals in the lab. Thank you.